Hello, Nick. We have Dr. Nicholas Ashton and Dr. Aitana Sogorb-Esteve. So I'm going to get your name wrong. And we should be getting joined as well by Dr. Amanda Heslegrave, who I'm sure will be dropping into the stream. Aitana, why don't you go first? Introduce yourself.
Yeah, sure. Well, so as you said before, Adam, I'm a Race Against Dementia fellow. I joined this wonderful team at the beginning of this year, and my focus, as I'm in this session, is fluid biomarkers, but I work in a specific type of dementia, which is fronto-temporal dementia. And it's a bit different from Alzheimer's disease, and well, it's a bit complex type of dementia, so the diagnosis and the development of therapies are quite complicated. So my focus is looking for these fluid biomarkers or blood markers that can help with the diagnosis, because to this date we don't have nothing that we can use so far that it's very specific of this type of dementia.
Thank you very much. And of course, you're Race Against Dementia fellow as well, and we're going to be joined by Race Against Dementia and the other charities at 4:30 today to hear about the importance of their work and funding. Do you all know each other, I should say?
Yes.
You all do. Okay. So now I'll come to Amanda. Thanks for joining us, Amanda. Did you suddenly have a panic when were you watching the live stream and then heard that you should be here?
Yeah.
Did you think it was
20 minutes and then all of a sudden I'll get a WhatsApp, but no, that's fine. So I introduce myself now, yes?
Yes, absolutely.
Okay, hi. Right. Hi, I'm Amanda Heslegrave and I work at the UK Dementia Research Institute and spend much of my time looking after running the Fluid Biomarker laboratory there, which incorporates the Biomarker Factory, which is a venture that we have been working on for a year or so now, where we set up a facility to help researchers within the DRI, but also throughout academia, both in this country and abroad, to measure their cohorts with the with the appropriate technology. We've got all sorts of different technologies, and I know that Nick will be talking about similar things. And so, yeah, so we've grown the lab to be able to offer these services to people and collaborations.
Thank you, man. That I've already got questions. But before I ask them, Nick, I'm going to come to you now. This is Dr. Nicholas Ashton, who is at the University of Gothenburg.
Yeah, hi Adam, thanks for the invite. So yeah, I'm Nick, I'm an associate professor at the University of Gothenburg, and I should spend some of my time at King's College London, but
are you saying you're not there often?
No, well, I'm not saying that. This is live, so I'm always
there all the time.
So my job is very in line with Amanda's and Aitana's, and mainly my work is in leading blood tests for Alzheimer's disease, and making sure that these tests are ready to be implemented in somewhere like Amanda's institution, which then they can be offered routinely for clinicians and pharmaceutical trials. So mainly this is phosphorylated tau. So this is a well-known biomarker in our world, and I think it's made it to the mainstream press now, knowing that we can actually accurately diagnose people with Alzheimer's disease through a blood test. So that's what I've been working on in the last 10 years or so.
So that's great. So I could be getting this wrong, because I think maybe Aitana is the one that I'm going to get this wrong. I'm thinking about how you all seem to kind of network together there. You've got Nick, you primarily focus on finding what are the best new, doing the discovery part as to what are the best new tests, and then Amanda, you're the one that has to kind of put them into use. To some extent, I mean, you're the kind of, you run the factory then that kind of does these, and Aitana, you make use of them. You're a bit more...
it's also more exploratory and not Alzheimer's disease. So I think we complement each other mostly.
Well, yeah, and I suppose I should say, I mean, we don't just work on Alzheimer's disease. Mm-hmm. We process samples for all kinds of neurodegenerative diseases, and not only in humans. So, you know, it's like it's not focused to that extent on AD, but a lot of what we do is AD, of course.
So I'm going to come back to you, Nick, because it feels like your work's at the start of this journey, if you like. Your work's at the start of this process, and then Aitana too. So what do you do? What are you working on right now? What's your research focused on?
So as I mentioned before, I think we have now tests. I mean, we have tests that diagnose Alzheimer's disease, and I think the biggest signpost for this is now, drug trials are using these blood tests to recruit people into their trials. They trust these blood tests. So multi-billion-pound drug companies are trusting these. It's essentially at the core academic tests that we've developed as a community in the science. So this is a really good marker that these tests are trusted. And now we're working on really making sure that these are ready for clinical use. So how can we implement and communicate this to a clinician at the primary level, and giving them the education of what this means. In Sweden, this is a completely different process as it is in the UK. There's a lot of different regulations. So this is going to take some time in some countries, but quicker in others. So that's where we are right now. But of course, this is only a very, very small part of dementia. And this is targeting amyloid, as we heard earlier, and tau. So these are the two pathologies, and many other types of dementia and symptoms and consequences of dementia that we do not have biomarkers for. And Aitana is specifically one about frontal-temporal dementia. But I hope what we can learn from this decade process of developing blood tests for Alzheimer's disease, that it's going to quicken. And we've learned so much on what works and what doesn't work, that we do find very quickly tests for frontal-temporal dementia and Lewy bodies dementia and all kinds of things as well.
Because that's one of the issues right now, isn't it? Is from the old-fashioned tests, the cognitive tests and things like that, you can say, yeah, you've got a memory issue, but actually then dividing that down, whether that's FTD or Lewy bodies or vascular dementia, Alzheimer's is harder. So are your tests at that point now? Can you take a test? How early on can you employ a blood test, and then how good is that at deciding which type of dementia, and how early on does that work?
Yeah, great questions. So they work maybe 15 years before you get symptoms if you have the pathology. Wow. Now, these are, at this point, they're not 100% accurate. So it's kind of like a triage process. You can say, okay, we think that you may be on the process of Alzheimer's disease. We need to confirm it. Now, at the symptomatic phase, when you come into a clinician where you have symptoms, I mean, the best versions of these tests are equivalent to our gold standards now. So we can almost,
So this is as reliable as getting a cholesterol test or a diabetes test or something you get routinely.
I think we're getting very close to that. I mean, we will be able to do that. I mean, of course, in a clinician sense, this all needs to be taken into context with the symptoms. So let's say Aitana sees someone with frontal-temporal dementia, and it's classic signs of frontal-temporal dementia, but their blood test for Alzheimer's disease comes back positive. This doesn't mean we should change the diagnosis. This means that there is Alzheimer's disease pathology contributing to the symptoms. So when someone has symptoms, it's kind of just another tool that a clinician has to come to a conclusion, but that conclusion needs to take into lots of different considerations.
And I'm guessing there's like a curve here, which is, you mentioned 15 years before, and the longer you get, the more accurate that gets. Absolutely, because the symptoms kick in. And so so clinicians use this in combination with some of the other things. I mean, today we've heard about all kinds of things. We've heard about retinal eye scans to look at, and we've got a podcast recording about coming up, to look for amyloid. And we've been talking about gait analysis and some of these longitudinal cohort studies that are capturing more information. So you still need those other things. Those tools can't all just be thrown away.
No, and I tell you why now a blood test is really, really important in light with lecanemab results that had this week and donanemab. I mean, a lot of people have said to us over the years, why would I want to test to know that I have Alzheimer's disease? And to some degree, I agree with them. But now this argument does not stand, because we are on the verge of having disease-modifying drugs. So at the moment, particularly in the UK as an example, in general, you come and have a neuropsychological test and maybe it looks like Alzheimer's disease. It's very rare that you'll get a spinal tap, and it's even rarer that you'll get an imaging, PET imaging, which looks for amyloid as well. So a clinician basically has no biological tool to say, is this person going to benefit from an anti-amyloid drug or not? Now we have a blood test which is available for everyone. This is what a clinician will be able to use to decide, is this person eligible and will benefit from this type of drugs or not? So this is why it's really important.
So Aitana, so do you use these tests that Nick's and his colleagues are kind of creating, or are you also making your own? Are you saying, right, okay, well, I want something a little bit different, I want something more specific?
Yeah, so for frontotemporal dementia, we can use the tests that Nick is developing and the ones that he has mentioned, but it will only tell us, it's what is called differential diagnosis. So it will only tell us that the person has not Alzheimer's disease pathology, but it won't give us any more information, or so nothing specific of frontotemporal dementia. And so my research, I think it comes in a bit earlier stage than Nick, in which I do exploratory research to look for certain proteins that are more specific of FTD and can give us a diagnosis tool similar to A beta and tau.
And then you can say, find me a test that finds this protein.
Exactly, yeah.
Nick, how many other people are there out there like Aitana who's saying, I need a specific type of test? You know, generic Alzheimer's test is no good for me. I need something special.
Yeah, hundreds and thousands of like this. And I think also we need to remember that we were spoilt with Alzheimer's disease, where we had already gold standard tests. So we had this brain imaging methods and cerebral spinal fluid methods, which have been kind of gold standards for Alzheimer's disease for decades. So we have fantastic research cohorts, and this has made it a lot easier. In FTD and other types of dementia, the gold standard is generally still clinical symptoms, and this makes it a little bit complicated to find blood tests for this. So we're still, maybe Aitana can correct me, but we're still looking for gold standards, which in living patients for some of these dementias, which is going to be the first next step.
So there are all these different stages to this. And Amanda, what happens with your stage of this?
Well, I'm just commenting on what Nick was just saying there. The gold standard, yeah, we've got CSF tests that are used, but not in so many places in this country. So kind of one of the things that the news about lecanemab and the revolution in blood biomarkers is hopefully going to push these things forward, because we're so desperately going to need the facilities to be able to test people properly, but also the understanding of the kind of frontline medicine that these things work and that they will be useful and we need to use them. So I suppose our research, by working in the research only arena, is giving lots and lots of information. We're measuring lots of different cohorts, sort of Lewy body dementia and FTD cohorts and AD cohorts, and all of this information is additive, but also working with people who are in right now putting together projects to measure phospho-tau in old age psychiatry clinics, and work with the primary care clinicians to see how that helps them to diagnose people, what they need to have to be able to, utilize these tests properly.
Because that can be massively beneficial. I assume that if you have say you already have an existing cohort of people, and then you have a new amazing blood test that comes along, kind of applying that to the people that you already had, who have perhaps had their diagnosis, gives you more information. It just kind of helps that cohort of the richness of the data you have about them to differentiate between the different types, different levels.
Yeah, and I say the thing, we get inquiries all of the time for the new phospho-tau assays that are coming out, which are not available to me yet. I should say that for anyone listening, but they will be.
Why not? Is it money?
It's not. It's tech? I think it's literally getting them off the ground, isn't it, into commercial usage. I think it takes an awful lot of work to put an assay from a research lab into a position where it can be commercialized.
And is that something that is that something that governments or funders could improve? I mean, we're always looking for ways to improve the system, drop out those delays. Who can make that difference? Is this just a slow process?
I guess Nick's got more experience of that than anyone else.
Yeah, so I see that academia and pharma need to work as a partnership, and academia really makes the kind of scientific breakthrough. So, I mean, in our lab 2 or 3 years ago, we were the first to say this is possible. You know, we can do this, and this is how it is. But at the end of the day, we're a university, and we don't have the input and the kind of manufacturing structure to put these assays together consistently and batch to batch variation. You know, how do you control all this? That's where, you know, a pharmaceutical company and takes this and makes it a finished product, and they need it as well for their trials. As I said before, they're using them.
And I suppose what drives them as to which one they focus on, or how much effort they put in, or how quickly they do it, depends on what the likely demand of it is, as to whether it's something they want or they can sell or use. Or
exactly, and it's also determined based on our research studies that we do with Amanda. I mean, they base a lot of their decisions based on what we report in academic papers and working with us to do this. So it does, in general, work as a very good partnership.
So are we at the point where we talked a bit about CFS with this spinal fluid? Are we at a point where your blood tests are as good as that? Do we need to, can we stop kind of with spinal fluid tests now, which we know are not very popular when it comes to these research cohorts? I was invited to have one myself, but in the trial I'm in, and I said, no, you're okay, thanks. Which I feel bad about admitting, but are we at that point?
I was just going to say that most people don't get spinal lumbar punctures in this country, I mean, nobody anyway.
Does, do they?
So I feel that something that we could use, like blood, you know, apart from that, and I think we can actually. I mean, we can with the phospho-tau is certainly good enough. The amyloid beta tests are getting better, I think, and we're working on newer versions of that, which are more sensitive, which hopefully will help in that area. I'm not sure if Nick knows what we're doing on that side of things with different forms of A beta, but I think that actually we're not using them in this country very much at all, not as much as Sweden for sure, and that the blood tests are just kind of a no-brainer, really.
I mean, so right now, I mean, the chance of the CSF and things like that are kind of only, as you say, used on rare occasions, aren't they? Not a part of the normal diagnostic pathway for Alzheimer's in this, certainly not in the UK anyway. I guess maybe in other countries where tests are charged and they're just, you know, they'll do all the tests. Go on, Nick.
Yeah, so I agree with Amanda. And having a blood test is basically going to offer you something to people that had no other option, actually. And that's a vast majority of people in the UK and around the world. One thing we just need to consider is that if you do take a CSF test from someone, you have a lot more things you can explore for the diagnosis. So you have tests for alpha-synuclein, which could potentially say that it's you know, a dementia Lewy body type of dementia. In blood, we can't really assess central nervous system inflammation, which could cause differential diagnosis for dementia. So CSF, if it's available and it's done routinely and it's going to be taken, it's probably still the best option. Not because the tests are better, you just have a better variety of tests.
So still has its place, and if you're invited to do one as part of a trial, you really could. And I'm guessing, Aitana, it's used more routinely with your work.
Yeah. For exploratory studies, you usually start with CSF because it's the fluid that will better reflect what is happening in the brain. So then the process is, if you find something that is very interesting in CSF, of course, we try to transfer it to blood. But as Nick said, there are some kinds of biomarkers that are not measurable in blood, or you have to be aware that they might come from other parts of the body. So maybe you are not able to detect any changes.
So for anybody listening, don't forget you can post your questions in the chat or use hashtag Chatathon. We're talking about CSF there, which is the spinal tap. This is a big needle that they put in your back, which is why you can understand people are more keen to have the blood test. Let's go back to blood tests now then. So we say that they're available, better for some things than not others, but they are there for clinical use, and their practical use right now is mostly I'm guessing, as a mechanism of finding the right people to participate in trials that are looking for very specific people, because quite often drug trials are looking for very specific people, because that's what the trial's designed for. And then as we progress more routinely, as different treatments become available, it'll be how this kind of test will allow you to decide which combination of treatments might be the right ones to use, at what time you give them to people, whether it's too early to give them to them or too late, or which different drugs for different periods of as your disease progresses. So it's not the question. I'm just answering my own question there, so I'll come up with one. So do you see a point coming where this is where blood tests will be offered as part of a routine clinical diagnosis pathway? Go on, Amanda.
Oh, I mean, yes. At the moment with the disease modifying treatments, we're right at the start, aren't we? What we've said is, yes, we can change the course of the disease, or we can slow down the worsening. That sounds a bit clunky, but I think that's what really has been shown. But that obviously will be a springboard to the drug that we can offer people that can be meaningful. And so we're going to need to have those blood tests available to GPs, etc., to be able to push those people towards that drug.
What do you think, Nick?
Yeah,
is this? I mean, you've got more. You mentioned you've kind of worked with different countries around the world and stuff. Is this already more used elsewhere in the world?
No, I would say in America this is a real situation now, because they have an approved drug in aducanumab, which hasn't really worked out. But I mean, you need to diagnose people now. But I do think that has clinical utility right now. So explaining the situation: someone comes into a clinic, the clinician thinks they have Alzheimer's disease. The phospho-tau test, which we trust a lot, comes back negative. Now this kind of then thinks to the clinician, all right, there could be something else at play here. So maybe they go for an MRI or an FDG PET scan looking at glucose metabolism, and this could quickly highlight that someone's dementia is caused by something different, like the fronto-temporal type, which you would highlight from an imaging scan. But the blood test there has quickly identified it, rather than a 1 year or 2 year process of trial and error.
Yeah.
Also in patient management, if you don't have Alzheimer's disease, you don't want to be on symptomatic treatment for Alzheimer's disease. Maybe this is actually it's like a primary psychiatric disorder, which just causes temporary cognitive decline, and they just happen to be in that state when they visit the clinician. A blood test would highlight this very, very rapidly. So, and
because the diagnostic pathway is so based on like 2 quick visits, and often it doesn't even involve an MRI scan, using this to be able to discount Alzheimer's, so you can keep looking rather than just to go, oh, they've got Alzheimer's, we'll just sit and wait and put them on a certain medication for the next 2 years, is discounting other things clearly very beneficial. Aitana, I feel I didn't give you enough chance to talk about the GEMFI cohort. Go on, you've got a minute. Tell us about the GEMFI cohort.
Well, so the GEMFI is an international cohort it has now more than 30 centers across Europe and Canada, and so between the 30 to 50% of people with frontotemporal dementia, they have a genetic cause, which is a change in your genetic code that is developing the disease. So in GEMFI, we recruit participants with this change, or it's called mutation, that goes FTD, and we do, so it's a longitudinal cohort, so we follow up all the participants, and we also have presymptomatic mutation carriers, which are people that are positive for this genetic mutation, but they don't have any symptoms yet, and symptomatic mutation carriers, as well as controls. So it's a very powerful tool for indeed fluid biomarker research, because we have a better understanding on which is the underlying pathology to those FTD symptoms, and it gives us more information that to maybe differentiate from Alzheimer's disease pathology and so on. So yeah.
And
It's
if there's
very-
anybody watching out there who's a carer or has been given a diagnosis of FTD, would you know about them already? Is there a chance that they wouldn't know about this? Is there anything you'd want them to know to say, hey, you should come and somewhere they should go to come and get involved in your research?
Well, I mean, we have a website on GenFi with all the information and all the centers across Europe and Canada, as well as some information for participants and carers about FTD and the genetic mutations and so on. So they can always contact in the website, and they can find more information there.
Brilliant. Thank you, Aitana. Last question. I'm going to run over time, but I think this is important because we've seen the media this week has gotten suddenly very interested in dementia and Alzheimer's disease. Or it's quite hard to attract the media to be interested in something, no matter how exciting it is. Do you have any pet peeves about the way blood tests for dementia are covered in the media? Because they have been talked about a lot in the past. Is there any? Do they get it wrong, or are they all right? Go on, Amanda. You've looked like you've got something to say. Go Amanda, and then I'll go Nick, and then we'll.
No, it's the media, isn't it? They've been saying there's a blood test for Alzheimer's for as long as I've been working in the field, and only now do I feel kind of confident that that's actually true. Happening. So, yeah.
What do you think, Nick?
No, I don't have any problems now with it, and I think that I agree with Amanda. I mean, ever since I've been working on this for 15 years, my PhD was in blood biomarkers.
Oh, sorry.
So I'm actually quite happy where the state is now. I remember I was at this AIC conference in 2013 in Copenhagen, and I was the only blood biomarker talk. I was on the last day at 3:00 in the graveyard shift, and no one cared about it.
And now you've got entire.
I come back into these conferences now with a smug smile on my face. But I think that the way that they are covered in general now are pretty good. I think that they communicate with the right people. Before we had blood tests which didn't work in some cohorts, and there were proteins that we weren't quite sure which pathology they were involved in, and even the academics didn't really understand how these tests were working then, and that's what made it very confusing. But I think everyone is on the same page now. Everyone's clear that they work and confident they work, and I think that's now communicated into the media.
There's real opportunities now. I think anybody, whether you're a PhD student or a researcher, if you're not enhancing the research you're doing by applying this blood test, you're potentially missing a real opportunity to collect some useful information and to really drill down into the people that you're researching. So you should definitely be talking to Amanda about how the DRI biomarker factory can help you and your research. And also as well, I think we should just say that if you suddenly go to your doctors tomorrow, you're probably not going to be asked or invited to do a blood test for Alzheimer's disease unless you go to participate in a trial. There is an opportunity. They might ask you then. But I think we might be close to seeing that more routinely used. I don't know if you all agree.
Mm-hmm. Yeah.
Thank you so much to my brilliant guests, Dr. Amanda Heslegrave and Dr. Nicholas Ashton from the University of Gothenburg, King's College London and UCL, and also Dr. Aitana Sogorb-Esteve, who's a Risk Against Dementia Fellow, one of the brilliant charities that will benefit from our work today. And of course, I'm guessing you guys are funded, you're in the DRI, aren't you? So that's also Alzheimer's Society and Alzheimer's Research UK that fund that work as well. Thank you all very much. It's fascinating. And we've got two podcasts. I should add this. We've got two podcasts. Nick and Amanda are both going to be hosting a podcast for Dementia Researcher in the next month about blood-based biomarkers and alternative biomarkers. So come back and listen to the podcast in January in your podcast app to hear them talk more about their work. Thank you very much.
Thank you.
Thank you.
Okay. 5 minutes late, but that was fascinating. I couldn't not ask those questions. But my next guests have been very patiently waiting in the lobby, and they are, over the next 30 minutes, I'm going to be joined by, turn on your caption, turn off the other caption. I'm going to be joined by Dr. Danielle Newby and Dr. Laura Winchester, who both work at the University of Oxford. And I gather you said when I was setting this up, you used to sit next to each other, but you don't anymore. Is that because, like, did you fall out?
Yeah, we fell out. I couldn't cope anymore.
You can't put up with, honestly, did she nick all the milk all the time?
Yeah, it was actually.
And the stapler, actually. The stapler was the final straw.
So over the next half an hour, we're going to be back. We've already had a talk about omics earlier today, and now we're going to be talking about genomics, big data to find new medicines and to help diagnosis. I don't know, this is going to be a little bit of a change, isn't it, Danielle? Because last time you here, your talk was finding teaching new drugs, old drugs, new tricks.
Yeah, that's right.
Right?
So it's teaching old drugs, new tricks. So looking at the benefits, and now I'm looking at like the safety. So people.
Now you've done that. that's done.
It's all done. No, I'm still kind of doing the benefits, but also looking now at safety as well, because that's got patient benefit as well. Before
we just go into this session, I'm just going to do my half-hourly reminder that today we're raising money for Race Against Dementia, the Lewy Body Society, Alzheimer's Research UK, and Alzheimer's Society. If you haven't already donated, please do consider giving what you can. We know things are tough right now, but every donation will make a massive difference and really help. You can donate at chatathon.uk/donate, or go to chatathon.uk and click the link at the top of the page. And if you're watching on Facebook or YouTube, you'll find a link in the chat below as well. Those charities really will put your money to great use to fund research. And as the guests that have just left before, the UK Dementia Research Institute is one of the organisations funded by Alzheimer's Research UK, Alzheimer's Society, amongst others. And that work wouldn't be possible without your donations. If you can't afford to donate, there's still different ways you can get involved. If you go to joindementiaresearch.nihr.ac.uk, you could potentially be involved in a research study yourself, even if you don't have dementia. Or if you go to the charities websites, it's raceagainstdementia.com or alzheimersresearchuk.org, alzheimers.org.uk, or lewibody.org. They've got lots of different volunteering opportunities. That could be to give advice on different trials, sit on trial steering committees, give your opinions on research priorities. It could be to run a charity event yourself, or just to be a volunteer. So there are lots of ways to get involved which don't involve giving out money, but if you can make a donation, please do. We'll just watch a quick 30-second film now from the Lewy Body Society.